So, good afternoon. I invite Professor Tushar Chakraborty to the dais. Professor Chakraborty, Professor Bhate, my colleagues, the invitees, and my dear students. It is a great pleasure to invite you for the KKG Menon Memorial Lecture by Professor Tushar Kanti Chakraborty. Actually, the KKG, I have not interacted much with Dr. KKG Menon. I have gathered the information only from this and Professor Bhate. And if you look at the kind of distinguished personalities who have adorned this podium under this KKG Menon Memorial Lecture, you will come to know the value of this lecture series. The very first speaker in 2003 was Dr. Mashalkar, and I think I will not run the entire list. I will say a few people: Dr. Ramarao, Dr. Vijay Raghavan, then whom I also know. Last year we had Professor R.K. Prasad, Dr. Shivaram, and so that speaks of the kind of place what Dr. KKG Menon had in the corporate research area. I think he is one of the very important successful researcher in spite of being in a corporate sector. And I had an interaction with Professor Tushar Chakraborty during lunch. He's a very thorough disciplinarian and a very good teacher in his own right in organic chemistry. So now the podium is yours. Before that, I want my colleague Dr. Saha to give a formal introduction of the speaker. Thank you.
Good afternoon all. It's my pleasure to introduce Professor Tushar Chakraborty to you. Professor Chakraborty had his early education at the Presidency College, Calcutta University, from where he obtained his BSc in chemistry. He got his master's MSc in chemistry from IIT Kanpur and his PhD in organic chemistry from the same institute in 1894. After postdoctoral studies with Professor K.C. Nicolau at the University of Pennsylvania, from 1984 to 1987, he started his independent career at the CSIR IIT Hyderabad. Then he rose to the position of scientist level G, and in 2008 December, he undertook the directorship of CDRI Lucknow and served there till December 2013. In January 2014, he joined IIT Bangalore as a professor in the Department of Organic Chemistry. After superannuation in July 2022, he is presently serving as the honorary professor in the same department. Professor Chakraborty's research interests span diverse areas of organic chemistry, from total synthesis of biologically active natural products to peptides and peptidomimetics, and designing new molecules for potential therapeutics applications. He has published many papers, guided several PhD students, and served as a member of many scientific committees, editorial boards, and advisory committees of many journals. Professor Chakraborty is the recipient of the SSB Bhatnagar Prize in Chemistry in 2002. He is also the fellow of all the three science academics in India: Indian National Science Academy INSA, Indian Academy of Science IAS, and the National Academy of Sciences India NASI. He is also the recipient of INSA Golden Jubilee Commemoration Medal for Chemical Sciences in 2022, Chemical Research Society of India CRSI Silver Medal in 2016, J.C. Bose Fellowship in 2008, CRSI Bronze Medal in 2002, Ramana Fellowship CSIR Young Scientist Award, and many more. So with this, let me welcome Professor Chakraborty, and I request him to start the presentation. The stage is yours, sir.
Is it okay? All right. Okay. Thank you, Professor, for your very kind introduction. And first of all, let me thank Professor Bhatte and Professor Shekhar for basically deciding to have me here to give this very prestigious lecture, Dr. KKG Menon Memorial Lecture. And I was really pleasantly surprised to receive this invitation. And then I looked at the eminent people who have given lecture before me. I was a little bit nervous, also I felt very humbled. And when I accepted this award, invitation to give this lecture. And the topic that I'll be speaking on today is in an area where we haven't done much as compared to other areas of expertise, that is total synthesis of natural products. And this is an area that we started working quite late in my career, much after I started my independent career. And the first paper that we published when I was well past 40 years of age. And we realized that age has no barrier in learning, so you can learn at any age. But some of the best works from my group we have published in this area. And so before I tell you about the work that we have been doing and its relation to COVID-19, let me also first pay my tribute to Dr. KKG Menon. So I was looking at his CV, and then I was amazed to find the kind of expertise he had and acquired over the years in diverse areas, starting from chemistry, foods and personal care products, human and animal nutrition, toxicology, pharmacology, agriculture, and some of these things possibly I'll try to touch towards the end of my presentation. And very early age he started his career when barely we finish our high school nowadays, at the age of almost 20. He was working on a plague vaccine. Like a vaccine is an area I have had some interest. And so, from that area, then he went on to do the PhD, the best university, one of the best universities in the world those days. And after a couple of postdocs in very famous institutes in the world, he came back. I believe he stayed here for, short period and went back again. And I think it was Dr. S. Varadarajan who was instrumental in getting him back to India to join Hindustan Lever, and where he worked throughout his life, rose to the highest level there. And I have not known him personally, but I was telling Professor Bhatte that I had opportunity to talk to Dr. Varadarajan on many occasions. So I used to see him often in IICT where I was there. I used to see him in INSA guest house in Delhi. Whenever I used to go to Delhi, every day he used to come there. And his son was almost like my contemporary at IIT Kanpur, who is now my colleague at the Indian Institute of Science in MBU department. So many of these people, when I look at their career, how much knowledge they have acquired in their lifetime and what kind of expertise that they had. And I'm sure in India, in the different institutes in those days, we had very limited facilities, not much as you have today. But in spite of that, their enthusiasm and kind of building the infrastructure or training students over the years. They really astonishes you how dedicated these people were. And I was very fortunate to give some of the memorial lecture, like I was mentioning, I gave the memorial lecture of K. Venkataraman, whose name you have this auditorium, a memorial lecture in NCL. I gave the memorial lecture in Professor T.R. Shesharji in Delhi University, Professor Ashima Chatterjee in Calcutta University, and what kind of people they were, how dynamic, and they built a not only excellent teacher, excellent researcher, but also the fantastic leadership quality. They trained so many generations of people and during their lifetime. And that's how these institutes were built in the country. And I'm thankful, I think, to Vardhanajan to bring back Dr. Menon to the country and lead an organization like Hindustan Lever. I feel it was a loss for the academic circle. As an academician, I can say, but it was a gain for Hindustan Lever. But we do need, very talented people for the industry, because unless you have good talented people in the industry, so especially today, if you say "Make in India," that will only remain a slogan. So you need qualified, scientifically sound people who are, holding the affairs in many of these industries. Then only you can compete with the world. Otherwise, we always remain only the follower. We can never be the leader. So in many areas of science, we only remain follower. So unless we have quality people coming who are very enthusiastic as they were at those times, and if they had, this kind of facility as we have today, I am sure, we would have been much ahead, than what we are today. So with these few words, I again feel, humbled and very fortunate to have my name associated with this very talented and very extraordinary person. So I go to my presentation. Before I start presenting, I must, admit that I have very limited knowledge in this area, because in my lifetime I have not done any biology course, and only little bit biochemistry I learned from a course from one professor during my MSc, Professor S. Ranganathan, and that's all. And then most of it, later on I learned after I joined IICT, and also during my stay in CDRI, because there I used to get surrounded by hundreds of clinicians, pharmacologists, and many other biologists, and they are wonderfully talented people. So I learned a lot of biology from them. So, but if there are biology students in the audience, so you will excuse me for my ignorance if I make any mistake. So now, why RNA therapeutics? Because if you see, so all the small molecule drugs that you use today, and they are mostly targeting the protein families that you can see, mostly enzymes or transporters, ion channels, and receptors. So roughly around 672 or 700, you can say, human proteins are targeted by all the FDA-approved drugs. So only limited number, 700. This little old data, few years old, but number possibly will remain the same, not more than that. And sometime, of course, the drugs that we use also target the bacteria or, like a virus that enter our body. But those numbers also are not many. So beyond that, people thought that there could be a way to control a disease by modulating the transcriptional and translational process using small synthetic molecules like a drug-like molecules. And this pie chart gives you an idea that this is the human genome, out of which 70% gets encoded into what we call non-coding RNAs. That non-coding RNAs are RNAs which are not making any protein, but they're important to make those RNAs. Those RNAs have a lot of important function in the transcriptional, post-transcriptional process in the gene expression. And out of that 1.5% of the genome that gets into the protein that we have in our body, out of that only about less than 700 of these proteins are targeted by these drug companies for various diseases, whether it is diabetes or whether it is a cancer or any other disease. So that amounts to only about 0.05% of the human genome. So there is a vast amount of human genome which is available for you to explore whether you can modulate those genomic, information to control some of the diseases that, which is very prevalent. And what is the, difference between DNA and RNA? DNA is, the blueprint of life, and it, basically it has a very well-organized structure, DNA. And there are many molecules known which you can target the DNA. The minor group of DNA is targeted by small molecules. This has been known for a long time. As compared to DNA, RNA has a more complex structure because RNA sometimes acts as an enzyme. RNA acts as a blueprint from the proteins. Basically, from there you get the protein synthesis. And so, it will be interesting to find, the binding pocket of an RNA with small molecules, because of its structural diversity, could be exploited to design small molecular therapeutics to bind to RNA. Now, RNA as a drug target being targeted now in cancer, cardiovascular diseases, many other neurological diseases and many other diseases. And beyond messenger RNA, because not only messenger RNA, these non-coding RNAs also are important. There are many, many microRNAs, long non-coding RNA. They are all called non-coding RNAs because they don't encode any protein, but they affect the expression of other genes through a variety of mechanisms. So can one control this non-coding RNA so that, that can become potential target for drug development? And what is important is to have this particular target. Which target you will be controlling? Because there will be hundreds and thousands of RNAs are made, non-coding RNAs, by our human genome. So you have to find out a target. So that target should be ligandability of this target. So that ligand should be able to go and bind to those targets. So the target should be well-defined target. Second is identification of the binding motif. So when you know the target RNA, where it is going to bind. In case of DNA, you know the major groups and minor groups you are targeting. RNA structure is more complex. So you have to know what is the binding domain of an RNA. And then third one is development of a robust screening approach that will identify drug-like molecule. How do do the screening? Every day, nowadays you have high throughput screening. You screen millions of compounds against a particular target. So how do you use RNA as a target to find out a good screening method? And there are many works that are going on in this area. And two of them I have shown here with Shovik Maiti's paper. And one of them is like you take the RNA. Once you take the RNA from the cell, you can identify. Biologists know how to do all these things, separate the RNA. And then you treated an untreated RNA. And then you express, there's a called differential expression profiling. And from there you can find out which RNA is getting upregulated or downregulated on treatment with your compound that you have given. And from there you can find out the target. Suppose you don't know which RNA it is targeting. So you do this kind of profiling. You take all the RNA, then do the expression, and then you take treated RNA, do the expression, find out which one is going down, which one is going up. And second approach will be like normally even for drug discovery we do the same thing. What you do is like a small molecule. Suppose you have a target molecule, you have prepared a molecule, you anchor it on a column, silica gel column or any other Cephalus column or something like that. There are methods to attach the molecule that you have synthesized with those columns and then pass a pool of cellular RNA through it. So if this molecule binds to those RNA, they will get stuck. Those which are not bound, they will be passing through. And at the end, that molecule which you have attached, it's called affinity chromatography kind of thing. So you pass the solution of the molecule now through the column. So it will now, elute out the RNA which was bound to the molecule, and then you identify the molecule. This is another, method of screening. This just like protein targets, you do the same thing for the RNA molecules. So recently, one paper was published in 2002. As you can see, they have identified some molecule from a library of about 50,000 compounds. against one RNA, which is called XIST, XIST, which is 70 kilo base long RNA transcript that initiates X chromosome inactivation. So they found that inside that RNA, there is a 431 nucleotide domain, which has a repeat unit of GC. So a lot of GCs are there in that one. And they, from this library, they found that one particular molecule which has some micromolar binding with this particular RNA prototype, XIST. And they have done some control experiment with other RNA which has those GC repeats. So it is very specific for this particular RNA, that is XIST. And what they have done is 50,000 is another way of screening. 50,000 compound mixture was equilibrated with the target RNA, and then target ligand complex was separated by unbound ligands by size exclusion chromatography. So this is very useful technique. Even in our lab we have done. Nowadays you can buy small Eppendorf centrifuge tubes which has 2 layers. So inside layer, if you put this bound molecule which has an RNA plus molecule bound to it, and many other molecules are there, small molecule, they are not bound. And then you do the centrifuge, small molecules will come out because there is a small filter, size exclusion filter process, small molecules will come out. What remains inside the Eppendorf is only which is bound to the RNA or DNA. So we have done it for DNA, so you can do the same thing for RNA. So this is size exclusion chromatography or centrifuge machines. You can do the separation of the compound which is bound to RNA. After that you do reverse phase HPLC, then you can isolate that molecule which really bound to XIST. And that's how they identify this molecule. So this is one of the examples I wanted to show, very recent one. Many other molecules which are known to act on RNA. For example, you use a lot of amino sugars, aminoglycosides, like kanamycin, and for use for a long time as an antibiotic. They bind to 50S or 30S subunit of the bacterial ribosome. And that's how they kill the bacteria. I will not go into the details. If it binds to this RNA, when it binds, then it comes out the non-conjugated transfer RNA, non-cognate transfer RNA will come, and you get a modulated protein. So if you get mutated protein or modulated protein, then that leads to eventually the death of the bacteria. And that's how all these molecules work. And many other small molecule drugs have been developed which bind to ribosomal site. And many others. You can go through this review and you can, they look like drug-like molecule. And they bind to RNA. Instead of the protein, they are binding to the RNA. And the targets of these molecules have also been found out. So now while antisense therapies, are being explored for many years, small molecule modulation of RNAs is a relatively new concept. Previously, people are trying to target RNA using an RNA-based molecule or oligonucleotides, synthetic oligonucleotides. They said, what's the point of doing synthetic oligonucleotides? Because they have the limitations. So can you not use a small molecule? What kind of small molecules that you can use? A drug-like small molecule. The aminoglycoside like gentamicin and streptomycin are known for a long time. So people get inspired by this kind of molecule. But one of the problem will be to get specific binding, because it's not difficult to find a molecule which will bind to RNA, because RNA is strongly negatively charged. So you find any positively charged molecule and that will go and bind to RNA, but you may not get a specificity because you have RNA all over the place, all cells, everywhere, you have RNA. You want a specific binding to a particular RNA, so that is a challenge. Otherwise, many of these molecules like gentamicin and streptomycin have a lot of side effects because they go on binding, in other targets also and create, many other, health problems. So now people are thinking, in case of non-coding RNA, which RNA are you going to target? So one of the options will be to target microRNAs, because microRNAs are very small and very easy to manipulate. And this is again, some of the review articles you can come. MicroRNAs they control the expression of the gene. You get the messenger RNA made. From the DNA, you get the messenger RNA. But all the messenger RNAs, don't go to the protein. Only some part of it goes, protein. So how does one block this messenger RNA? These microRNAs go and bind to certain section of the messenger RNA, inactivate their expression. So, it will not be expressed into the protein. So these microRNAs also come from primary microRNA, then pre-microRNA, and finally dispersed with the microRNA. They are very small, almost like only 22 nucleobase long. So not very big enough. Very easy to synthesize nowadays because you have the automated machine, you can make those microRNAs. And if this microRNA in your body, it is the normal. If it goes up, there's a problem. If for some reason their expression is low, then also it's a problem. So it leads to diseases, and many of them, you can see the cancer, it leads to disease, you have a cardiovascular problem. So people have been trying to modulate, microRNA so that you can control all these diseases. So now coming back now to our work, long back, as I said, like after I joined IICT, one fine day, our director, Dr. A.V. Ramarao, so he called me to his office. So he said, what do you want to work on? And I said, I'm trained as a synthetic organic chemist. I will do synthetic organic chemist because all of us, we want to only remain in our comfort zone. We don't want to explore, other areas. Then he said, so-and-so is expert in carbohydrate. So maybe you should not do carbohydrate. I said, I also thought of working on carbohydrate, but okay. Then he says, so-and-so has expertise in this area. You can't work in this area. I was a little taken aback. Then he said, he's the director, so I can't do much to him. So then he said, why don't you start working on peptides? I said, I have not done any work on peptide. But, then he gave me one big, monogram prepared by one Professor Shivanandaya. I didn't know him at that time, but he was very well-known peptide chemist from Bangalore University. So then I went through that, monogram and found review article kind of thing and see what are the peptide work being done in India. And I sort of like a volunteer, I took it up as a challenge. After 1 or 2 years, I went to Russia. Those times we had Indo-USSR ILTP program. So it was still in Russia, so not, sort of like a USSR. So one of the best institute there, Shimak Institute of Bioorganic Chemistry. So I went there for 6 months. So I had the hands-on experience. For any work you want to do, you must have your bench knowledge, bench work experience, hands-on experience. So I learned, how to make peptides. I was working on malaria vaccine. So basically, you make the antigenic site of Plasmodium falciparum, that is like which causes malaria. It's called circumsporozoite protein. I will make the peptide by solid phase synthesis, then anchor it onto the protein, and then it has to be injected on mice, and you find out what is called antibody titer value. So only the animal part I could not do because I was always afraid to touch, work with animal. And at the end, you have to sacrifice those animal to take out the spleen and find out, how much antibody is getting generated. But I knew the subject very well after working there for 6 months. Came back and then I didn't do much work because I found that, peptides any person can do. Nowadays, you don't have to know any chemistry also. There's a machine, you give the sequence, machine will make the peptide. So then I thought, as a synthetic organic chemist, I should have some, signature in the work that I do on peptide chemistry. So eventually, we come across with a molecule called sugar amino acid. This kind of, you can say, hybrid of carbohydrate and amino acid. So basically, it's a hexose sugar, 2,5-anhydro, you can say. And you can start with any sugar. You can start with mannose, you can make the corresponding sugar amino acid. You can start with galactose, you can make the corresponding sugar amino acid. 6 position is converted to an amine, and 1 position you have an acid. So we started using, and slowly, many groups in the world started working on sugar amino almost simultaneously. Professor Fleet from Oxford University, Professor Kessler from Munich, and one group from Belgium. So, and also many groups in India started working. It became very popular, topic. And this is the first work I published. As I said, I was past 40 at that time. And what happened, we just took enkephalin, and enkephalin to amino acid, glycine was replaced by this sugar amino acid. Enkephalin binds with the delta opioid receptor and works as an analgesic, basically. So in that, within the cell surface, it takes a particular conformation as shown here, where the two aromatic residues come close to each other. And this was kind of pre-introduced by us because of the sugar amino acid, and it showed very good activity without much of side effects and other things. We did the biological testing. And this was because of a peculiar hydrogen bonding we shown for the first time, where hydroxyl is forming an hydrogen bond with an NH. Normally, hydroxyl in a peptide, which have a serine and threonine, they form hydrogen bond with the main chain, amide-linked protein, which is a carbonyl, where hydroxyl acts as a donor and carbonyl acts as an acceptor. Here, hydroxyl oxygen is working as an acceptor, NH is forming the hydrogen bond. So this is very unusual, reported for the first time by us. And later on, we found out that any 5-membered ring, whether it's a carbon, nitrogen, or something, if you have hydroxyl and carboxyl, if you make a peptide, it forms this kind of hydrogen bond. So what we proposed in 1998 on the basis of NMR, this was proven by the Belgian group. They used the same amino acid, put in some peptide other sequence, but they could get a crystal structure. And they proved the existence of this kind of hydrogen bond. So we use this work extensively for making all kinds of molecules, as shown here. Enkephalin analog, then some cytosymmetric reverse stand mimetics. Many of them we have tested for HIV protease inhibitory activities. Cyclic molecules, this was prepared by cyclodimerization and all. Some of them we went on to use as G quadruplex ligand, then foldamers to organogels, and many other molecules as shown here. So basically, we worked on small peptides and foldamers of sugar amino acid containing cationic antimicrobial peptides, then G quadruplex binding ligands, and fully amide-linked RNA mimics for antisense therapies. So this is like a few slides on each area I'll show. We are interested in cyclic molecules. This is a cytosymmetric molecule. It becomes easier for the student because you have to make only one half of the molecule and dimerize the molecule. So gramicidin A is known for more than 80 years now. So A stands for Soviet. And they thought it will replace the penicillin because they have good antibiotic activity. During the Second World War, this was discovered. But this never became a drug, oral drug, because it has problem that it was killing the red blood cell also. So hundreds of papers have been published by people to make the analogs of gramicidin. In our lab, what we did, we replaced the DPro, that is in two ends, you can see in yellow color, we replaced by a sugar amino acid that's shown here. So you can see there are 3 chiral centers in this molecule. My student make all the 8 possible isomers of the analog of the cyclic molecule. Found out the best activity was found by this particular terminal, which mimics the stereochemistry of the diphenylalanine side chain, as shown here. And it also showed very selective anti-tubercular activity. Now, during the process, my student also made, this is a 30-membered ring. So 24-membered ring molecule, as shown here. He put some thiol also at 2 ends, because we found that with that you can attach to gold nanoparticle, and that reduces the toxicity even further. So you can attach this ligand to the gold nanoparticle and use that for doing antibacterial activity studies. But during that process, we found that gramicidin A also interferes with the RNA polymerase binding to DNA and inhibit the transcription. And then it was also shown that GS, gramicidin DNA complex, mediated high efficiency transfection of adherent mammalians. So this is the cationic antimicrobial peptide. We thought, why not we test it for binding to RNA? So this was done by Shouvik Maiti, and these are the 3 molecules. He, basically profiled the 96 therapeutically important microRNAs associated with cancer. So all the 96 were profiled, found out 2 particular microRNA, that MIR106B and MIR106A. They are being controlled by one of the particular isomers of this one, that is CP1, that is have a 2,5 trans stereochemistry. This is the structure of the CP1 on the top. and dose-dependent studies were carried out, and you can say out of that two, only MIR 106A was found to be affected by doing some dicer assay and all those things. I will not go into the detail. But important thing is out of that 96 RNA, only one particular RNA was getting affected by this particular molecule. So we are very enthusiastic about it, that it has a selectivity. What was the cause? What is the binding domain? We don't know at that stage. So one thing leads to another. So we, as I said, like we made a lot of cyclic molecules also, like this furan-based amino acid, and prepared the cyclic, and this was tested by Souvik Maiti's lab, again in stabilization of the G quadruplex. Like all our chromosomes at the end has a telomeric region. Telomeric region has a G quadruplex structure. Every time cell divides, this telomeric region gets shortened, and slowly it leads to the death of the cell. Where it does not happen is in the cancer cell. Because cancer cell, these cells don't die and then expand, continuously. So one of the enzyme involved in this one, telomerase enzyme, which controls the length of the telomeric region and does not make it go shorten every cell division. So people are trying to find out ligands which will bind to G quadruplex to make telomerase, inhibit the telomerase activity. So this is one of the natural products which was isolated and very active against, like a G quadruplex. So we are mimicking this structure. And synthesis as shown here that we do a lot of, dimerization, cyclodimerization. So when you have to make a symmetric molecule, we can use a monomer. It's like making a polymer. Polymer chemist will know. So you use a monomer, we use the same condition, but after making dimer, trimer, tetramer, it becomes thermodynamically favorable for it to undergo cyclization. So you don't get much of the polymer. You get almost like a 60%, 50% yield of the cyclic product. Otherwise, you have to make a linear tetramer, and then only you can cyclize to get this compound. In this case, since it is unsymmetrical, so you have to make a dimer, and then head-to-tail dimerization and made these molecules. And many work was done, G quadruplex binding and all those things, very good activity, 50 micromolar. And Souvik was sitting on it. I said, what happened? Souvik was our student in the department, so I could, sometimes shout at him also. He's now director of IGIB. So I said, why you are sitting on this? By that time, Shankar Balasubramaniam from Cambridge, he published a paper. I said, Shankar has published, I will not be able to publish that paper. And invariably, when we sent to JAX, it was not accepted, but finally, Souvik, we published it in Journal of Medicinal Chemistry. So their activity was much worse than ours. 50 micromolar, his was possibly more than 150 micromolar binding. And many other studies was done by Souvik, including zebrafish, where the development of the embryo was shortened by adding this molecule because it binds to a G quadruplex region or the promoter region of a gene called N-cadherin. Similarly, like a c-MYC gene, in cancer cell, many of these proteins get overexpressed. I don't know why, but everything goes wrong in cancer cell. You find, only half a dozen or 1 dozen proteins are, unusually very high concentration, telomere, c-MYC gene, many other genes. So c-MYC is one of them. Its promoter region has a G quadruplex structure. When we added this compound, that c-MYC expression was down-regulated by 90% in cancer cell. And one thing leads to another, as I said. So we are working on this. Then Shantanu Chaudhary from IGIB, he was also our former student from IICT. So he said, Tushar, can we do a specific targeting of this ligand to the gene? Because you have G quadruplex structure everywhere. So what we did was he gave the sequence of this one. Of course, we are purchasing this. We are not making this single-stranded DNA. It has one cyclic group here as shown here, and this where we have to target our M3. So this is now even more difficult to synthesize because you have 3 of them, but one of them has an M3. Not very difficult. You have to make a linear one and cyclize. And then that Caroline Batrozzi got the Nobel Prize in 2022 for this work, which is called strain-promoted click chemistry. And it works wonderfully because it's different than what chemistry my students do. These just put them in water, reaction takes place. You don't have to monitor, no TLC, nothing, no monitoring of the reaction. After half an hour, Then again, you can do size exclusion, work, because you use excess of the ligand, you can remove all the ligand. But so to make sure all the DNA that you have taken has reacted to it. So conjugation was done. And, but as a chemist, organic chemist, I was little reluctant to let it go without any identification. So we had to get it done by Rockefeller University. One person I knew got the mass spectrometry study, so it shows the same mass exactly as the conjugated mass. Also, you can see the HPLC pure. So this is the starting one, and this is more polar, coming faster in the HPLC. And HDS gel also it showed. But by the time student left, he did not make enough. We could not publish any papers. So still, possibly some studies were done in Santadu's lab, but I don't know whether he will ever publish or not. And so now these are some small molecule targeting the RNA. How about the oligonucleotide? Can you use oligonucleotide? Yes. If you have more of the microRNA, so you want to lower the level of concentration. So use anti-microRNA, which will go and bind to this microRNA, and so the total concentration of microRNA will be less. And if you have less microRNA, you can make the same sequence, not the anti-microRNA, just microRNA you synthesize and then give it to that. So one of them is called anti-MIR, anti-MIR, and then miRNA mimics a synthetic double-stranded RNA. So you can, whatever you want to do, you can prepare those molecules. But it was not very easy, because after almost 30 years, the first siRNA-based drug was introduced in the market, because delivery becomes a problem. Stability also a problem, delivery becomes a problem. So they prepared some, like a galactosamine encoded liposomal delivery system, and because of that it became, like a active, and it was delivering the drug into the hepatocyte. And then because it has a GalNAc molecule, and hepatocyte has a lot of, lectin, so it goes and specifically bind to the hepatocyte for a very, very rare disease. But not many drugs are very successful. So what people think, like a unmodified oligonucleotide having poly-on backbone are enzymatic susceptibility to poor cellular uptake, because they are not stable if it is a phosphate linked. And they have limited tissue distribution, rapid clearance. So what people are doing, you need to have a high binding affinity for selectively binding to RNA, target RNA, high endonuclease resistance, so it should be stable so that it does not get degraded very quickly, metabolized, and then cell permeability. So we had interest in getting, what are the changes people are doing? They are doing in the base, nucleobase changes, they are making the ring changes, and then sulfur-linked, phosphate linkages, and also like a peptide nucleic acid. Nielsen from Denmark has done a lot of work in this area. In our country, Professor Ken Ganesh has done extensive amount of work in peptide nucleic acid. In Calcutta, Surajit Sinha cultivation, he does a lot of work on morpholino base, like not a ribose sugar, but different, morpholino based polynucleic acid. Delivery also important, liposome and dendrimer and many other delivery systems have been used. Our interest to have a amide-linked dinucleotide and their incorporation in oligonucleotide. A lot of work have been done, for last 30 years or so, but mostly they have concentrated on thymidine kind of base, because easy to get. You can know that this AZT. AZT is azithromycin. For HIV, this drug was introduced. So you can buy commercially available molecule which has an N3 in the 3 position, make the corresponding amine, and then you can easily use. So most of the design people have used are based on thymidine and many other, like a guanidine connected one molecule here, and so on. Our interest to make a same azitid thymidine-based design. So we cyclized it and did not get much of activity for these molecules. So cyclic dimer was prepared. Then it took some years for my students to prepare all the nucleobases with carboxylic group on one end and FMOC in the other. Idea that you can make a solid phase synthesis using the peptide chemistry. So later on, I think Baijanti Kumar from NCL, she was working with Ken Ganesh group, and she also published a paper on the synthesis of some of these molecules following a different strategy. We start with glucose diacetonide, and following a very standard routine chemistry, although it takes 6 steps, you can make a common intermediate and then put all the nucleobases and then some protection, deprotection steps. Total about 10 steps, not very difficult. You have a good student, you can make at least gram quantities of this material in FMOC and CO2H. And using these molecules, during this time, Eric Rosner, in one of the big groups in what university? Binghamton University in New York. He has done a lot of work in this area. So they found out that if you use this amide-linked nucleobases, it's stable and it binds to RNA and DNA very well, and it does not affect the overall function of these molecules. And actually 2 of my students work with Eric Rosner group, but they do a lot of biology, and mostly they use 1 or 2 substitution in the phosphate linkages. So mostly it is phosphate linked RNA or DNA. 1 or 2 places they will introduce using the nucleotide solid phase synthesis method. And so of course we have to do, after you put the amine and all, you have to do structural studies. You know that RNA has something called C3 prime endostructure or a N-type structure. North and south you will remember. So if the C3 prime goes up, like a map, it is north. And if C3 prime comes down, it is called south conformation. And some people call it C3 prime endo, and this is called C2 prime endo. And but DNA has a mixture of structure. RNA is mostly like a N-type structure, like a north-type structure. And if you have that structure, then it is better, it will bind to DNA or the RNA counterpart. So we did a lot of conformational studies with this molecule that we prepared. I will not go into the details of the dimer. And this is the pseudo rotation wheel, it is called. On the top you see it is called 3T2 in the group theory term. And this has like a 3' endo structure. And at the bottom you can see the 2T3 kind of thing. The 2' position is up. And so from the NMR studies we could find out that they all go to the right hand side corner, almost like an N-type structure, and comparable to the natural guanine phosphate, adenosine phosphate, and so on. And then in water they showed even better, because this is the like a N type percentage. It's like almost like you can say 100% concentration of the N type. And you can see that this is the conformation. If you go teach your conformation to the student, use this, what is called this projection, what is it? Fisher projection, not Fisher projection, Newman projection of the structure. So you see the dihedral angle. This dihedral angle is 90 degrees. So you know from the Karplus equation that if the dihedral angle is 90 degrees, you don't have any coupling, zero coupling. So that's why the C1H was coming as a singlet in most of the molecules. So it shows that it has 100% N-type structure. Then we did was like try to find out whether they form any duplex type conformation or not. But during a lot of NMR titration studies, we didn't find any presence of any duplex. And then stability is also studied by treating with acid. This has to be, sugar molecule has to be strongly bound to the base. If it falls off during a physiological pH also, it will not be good. So some stability studies had to be carried out. It was little less stable compared to the natural one, adenine and guanine. But otherwise, like adenosine and guanosine. But because of more of N-type conformation, we are confident enough that this molecule possibly will go and bind well with the RNA or DNA counterpart. And we also prepared some cyclic molecule as shown earlier, because this also plays very important role in the bacterial life. Variety of bacteria like regulation of cell cycle and virulence and biofilm formation. So people are using the cyclic dinucleotide analogs to inhibit the formation of the biofilm in the bacteria. So they can act as an antibacterial compound. And a lot of variations were made. People are making this kind of ligand by click chemistry, by amide and clink, and this kind of urea linkages and so on and so forth. So my student, what they did, they made the linear dimer and tried to do the cyclization. Along with they also got the tetrametic compounds. cyclodimerization product, but took this one cyclic dimer and then deprotected the compound and then put the bases. But mostly we got the pyrimidine bases. Purine bases is actually involved, cyclic di-GMP, cyclic di-AMP. So you have the pyrimidine bases there, purine bases. So he got the only of the pyrimidine bases. Anyway, so we went ahead and did some antibacterial study, but they did not show any antibacterial or antifectal activity. The crystal structure of these molecules are shown here. And as I said, like this did not work with the preparation of the cyclic di-AMP or cyclic di-GMP analog. And then, of course, we are interested to make whether we can use them for making any sequence of the RNA by doing solid phase synthesis. So the difference between here and the other RNA molecule will be the phosphate links are all replaced with an amide link. And again, for that, we had to develop a method, a resin. For example, if you use ring resin, that requires 50% TFA to deprotect at the end. And that was removing all the purine bases. So we had to find out some other resin. And finally, it was found out that if you use cyber resin, then with only 1% TFA, we are able to deprotect this molecule from the resin intact, get the good mass and all those things. And later on, another student of mine prepared this sequence. This is MIR21. It's a microRNA reported to be associated with multiple cancer-related disease, cancer-related proliferation, apoptosis, and so on. So inhibition of miRNA is shown to, sort of like curing the cancer, at least in cell-based assays. And most of these microRNA, they have, all of them, something called seed sequence, which is very short, only 8 nucleobase. So when microRNA goes and binds to mRNA, they use this seed sequence to bind to the complementary mRNA sequence. So I thought if we make seed sequence anti, like an anti-MIR21 seed sequence as shown on the top, maybe it can be tested whether it can block the binding of the microRNA 21 to the mRNA. But student made again very small quantities and he had to go because he was doing only an MSc project, but this was prepared and maybe, someday some other student comes. I don't have very many students now who can make a little bit more so that some biological testing can be done for these molecules. So I don't have many students. Most of my students, they want to work only on synthesis. So very few students are there and they like a volunteer to work in the area of peptides or related subjects. After they leave, they say, sir, we could have done this, we could have. I said, when you are a student here, you are not doing this. All these ideas were not coming. I was forcing you to do this. But when they go to any Krugner group or some other group, that we could have done so much of work. So I have only one student who is now, we are working on something called nucleobase amino acid. So PNA has become very popular. A lot of people use, for gene silencing and all those things. But one of the problems is PNA does not have any secondary structure. So that's why this area is getting developed, nucleobase amino acid. Basically what we do, serine or threonine, you can replace the hydroxyl with the nucleobase. But because it is an, like amino acid based design, so it can have a beautiful tertiary structure, well-defined structure, which can go and bind. So now my only one student, she is preparing some of these cyclic molecules. If she is successful, we can test possibly some antimicrobial studies and other work. So this is where we stand. Looks very promising, but very difficult to find student who will be, taking up this challenge of making this kind of molecule. And now I come to the last part of my talk, for the COVID-19 association and the, RNA therapeutics. Because that all over the world, including the organic chemists, they were working very hard to find out the drugs which can control this disease, including, people who are working on vaccine and people who are working on many other, ways to mitigate this unprecedented, disaster to the human being. And all of you know about this disease, how it progresses. It was filling the lungs of the people with the fluid, and people had difficulty in breathing. Their oxygen level was going down because the iron, gas exchange was not taking place. You bring carbon dioxide, you take the oxygen in your lungs, but this was getting filled up, and it was pathetic to see people gasping for air. And it was very quickly, the structures were determined for all the proteins because of the cryo, EM methods and all those things. Today, within no time, within a month, people knew the sequence, and within no time, people knew the three-dimensional structure of these proteins, various proteins involved in the disease. Although it takes time, to make a drug, it requires sometimes 15, 20 years. But within few years, you had drugs because people have been working in this area for a long time. Same thing happened for HIV protease also. When it came, people are working on protease inhibitors. So some of these molecules were repurposed to make new drugs for the HIV protease. Same thing happened here. So people are working for many of those drugs before. They quickly translated that technology to making the drugs for COVID-19. And various places you can target. You have almost like a 30,000 nucleobase long. It's not very big. It's a RNA virus because it makes its own copy of RNA. It does not go DNA. HIV, for example, it has also RNA vaccine virus, but it goes to DNA. So it uses reverse transcriptase to make the DNA and in our body, and from the DNA again, transcription makes the RNA, copying the RNA, and virus gets proliferated. But this one makes the copy of the RNA by itself. It has a protein in itself. It's called the RNA-dependent RNA polymerase. So it makes a copy of the RNA once that RNA is there in your body. And various targets. For example, you have heard the name of the drug called remdesivir. So it blocks RNA-dependent RNA polymerase. You have a drug called Paxlovid, who is the main protease of the enzyme. After the whole protein is made, the main protease cleaves the whole protein into small functional protein that was required for COVID-19. Then you have the vaccines against the spike protein, which you have seen the picture, how spike protein, through that one it goes inside our body. And there are also some drugs which works on the host targeted agents. For example, when it enters our cell, it also uses some of the enzymes present in our cell surface, and so that can also be blocked, like a S2 inhibitor and JAK inhibitors and so on. Some of these, molecules like a cytokine inhibitor, 11-1 is JAK inhibitor, then 14 is glucocorticoid. They have led to a lot of side effects. People have thrombus formation, thrombosis, and many other related diseases when they are treated with this, immunomodulators or many other drugs that were given. But anyway, so some people possibly got relieved. A lot of people were saved because of the development of the drugs. You have heard of the vaccine also, so many of you have seen. So this slide was prepared much before she got the Nobel Prize, because I used to follow her work. This Kathleen Carrico, she was in the same university when I was at the University of Pennsylvania. She had difficulty in getting her grants, accepted. Because this is an area people are saying not good enough, but she kept on working. Her papers were getting rejected. She sent her first paper to Nature. Nature said it is something like a incremental increase of the knowledge. She said, I come from Hungary, I don't even know English that well. I had to look at the dictionary, what does incremental increase of the knowledge says. But eventually, I was talking to my daughter, she said, Nature should be given a Nobel Prize for rejecting many of the papers of the Nobel laureates because of the cynical view that they have. But anyway, what she has done, she has replaced the uridine with something called N1-methyl pseudouridine. You can see the nitrogen has gone up there in the 3 position, and carbon has come there. So it's a C-glycoside. It's very stable. So because the RNA-based molecules were not becoming, good drug because of the removal of the uridine in the system, it was. So she tried to make a stable version of this one, and this is taken up by the RNA-dependent RNA polymerase. So it can be copied without any problem. It is considered as a uridine. So body does not detect it as a new nucleobase. So with that only the success came. What they have done, these companies, I don't know, you can see Moderna had 3,819 nucleotide long, and Pfizer one was like approximately 4,200 nucleotides. So they are making the DNA. Basically, you have to make the spike protein in your body so that antibody will be generated against the spike protein. So you give the protein, or you give the DNA, or you give the RNA. So they decided go for the RNA. And so they were making the small DNA synthesis by oligonucleotide synthesis, 50 MAR or 100 MAR. Then all these about, how many? So 30 or 40 or 50 of them. Then you use the DNA ligase to make a big DNA plasmid kind of thing. And then you can multiply that in large quantities using the bacterial system. And then once you get enough of DNA, then you can use the enzyme and with the different uridine and other nucleobases to get the RNA vaccine. So this is how this RNA vaccine was made. Small molecule drugs also introduced. This is called remdesivir, and then favipiravir, molnupiravir, and so on. And many other, clofazimine, it was an anti-leprosy drug. It was repurposed to work on the S protein of the viral. Ivermectin was used, and this chemostat is an anti-cancer drug. This is also another enzyme inhibitor. There is one very successful drug came from Pfizer, made billions of dollars from this molecule. It's called Paxlovid. It's basically a tripeptide, but they modified it so much you can almost can't recognize it's a tripeptide. So in the proline, they have attached the cyclopropyl group, and this side they have trifluoromethyl acetate, then tertiary butyl group introduced here, and so on and so forth. So, but they have problem, and chemistry also very difficult, not easy. So all this process chemistry, even in India, people are copying this and then making these drugs available in the country. So large number of steps are involved. You can see this molecule has a chiral phosphate here. So you make this molecule by all these steps. At the end, you have to use this, like a nitrophenol-based, living group, and then this SN2 displacement takes place. You get a chiral molecule. And people are developing a lot of chemistry, because now next slide will show that you don't even need this chiral phosphate, and you, in presence of this chiral, catalyst, you can get this in one step, with a very high diastereosyl reactivity and 96% conversion. So, but the problem with these drugs, of course, they have now helped us to come off from the COVID-19. For example, I talk about molnupiravir. Molnupiravir has this molecule. This nitrogen NH2 has been converted to NHOH, hydroxylamine. So you understand that this can undergo tautomerism. So all of you have heard tautomerism. So this tautomerism makes it in an oxime form. So normally if you have an amine, amine does not form a double bond NH that easily. But once you have a hydroxyl, almost like 1:1, you have the what do you call a hydroxyl oxime form. And now when this is taken up by the virus to make its copy, so when you have this pool in the solution, it can be picked up at any sequence, because the progression of the copying of the RNA does not stop once it is introduced. So you can have a third position, this one, or another again in the sixth position, or an eighth position. Now if it remains in the hydroxy form, I am putting hydrogen here, then it can, the next copy when it makes, because what is it called? This is the cytidine. Cytidine G couples, C couples with G. So G will come. But if you have in the oxime form, it forms other hydrogen bond, A comes. So this is a mutation. So as soon as, in the mother, like a sequence, you have this NHC is taken up, when it is getting copied, it can be copied as a G or it can be copied as a A. So number of mutations are coming up there. And again, if G comes, next one will be C. But if A comes, next one will be U. So a lot of mutations are happening when it is being taken up by the system, the virus. So virus, people thought that if a lot of mutations come, virus will become inactive. But you today see what is Gen.1 sequence is coming. It has hundreds of mutations, but it's still spreading. Some people say it does not even use the spike protein to enter our body. It is using some other protein to enter our body. Spike protein has been completely mutated in many of these sequences. Now if you see, Next paper, so I am getting paper few months back, Nature, 2023. They have, studied the millions of SARS-CoV-2 genome sequences from people who have been treated with this drug called molnupiravir. People got cured, they were good elderly people who are given this drug. It was a very good life-saving drug for them. But what happened, the mutations passed on to other people. And, sometimes there are more than 100, molnupiravir treatment got transmitted between individuals who had accumulated the mutations. And if you see the mutations, this is the spike protein region. And large number of, mutations happening in the spike protein. That means in spite of spike protein mutation, either it is binding even better with our receptor, or it is becoming ineffective, or some other protein is, binding to our body and entering that one. And so it is spreading all over the place. And other problem associated with it, they say that it remains in the cytosol, does not go to the nucleus, okay, these drugs. But there are some enzymes in the body which is deoxygenating the 2-hydroxy group, because these are the 2-hydroxy group. Then it is entering the nucleus and it can get into the DNA. So DNA, it becomes suicidal for the body, because it should not go into the DNA making machinery of our body. And so people are very, very worried all over the place about the long-term implications of these molecules. Safety profiles of mRNA vaccine, for example, this is the small molecule. I am talking about this RDRP, that's the RNA-dependent RNA polymerase inhibitors. But even vaccine also, this what they call is nucleoside modified synthetic mRNA. You have seen the pseudouridine was used for the mRNA vaccine. It's getting accumulated in our body. RNA is very short-lived in our body. DNA lives for thousands of years. DNA does not get degraded, it remains in the nucleoside. The only difference between DNA and RNA is the DNA, you have ATGC, AUGC. U does not have the methyl group. T has a methyl group in the 5 position. That makes the DNA very stable, very different structure, stable. I mean, possibly how it has happened and all those things, evolution has taken care of all these things. But RNA, sometimes only 10 minutes within the body. mRNA is made, work is completed, and you don't know how fast the protein gets made in the body. In 1 second, in E. coli bacteria, 40 amino acids are getting added. Not 1 minute, maybe 1 second. Before you can even bat your eyelids, big protein gets made by E. coli 40 amino. So my student, if they have to make one dipeptide, they take 1 day. You have to make a dipeptide, you have to do chromatography, purification, and then collect. But the enzyme makes it so fast. So mRNA, most of the time, at the most, it will remain alive in your body for 20 minutes, 15 to 20 minutes, even less. So, but these mRNAs that we are using for the vaccine is living for a long time in the body, because it is not getting degraded by the enzyme because of the presence of the synthetic, nucleobases. And as a result, so people are worried about all this. Susceptible individual cytosolic impeded accumulation of NMS reverse transcribed DNA molecule activate RNA sensory pathways with an increased risk of DNA damage, chronic autoinflammation and autoimmunity, and even cancer. And so what happened, this is because of, N1 methyl pseudouridine, as what, she got the Nobel Prize for, which of course was a breakthrough experiment, because the first time we got the RNA-based vaccine, we got first time the DNA-based vaccine, because most of the Indians, we have taken Covishield, which is based on DNA. Even that also has implications, because, some of these places, okay, this NMS modified, nucleoside modified synthetic mRNAs, post-marketing studies have shown that these mRNAs are passing on to the breast milk. So then, it goes into the adverse effect of the breastfed babies, long-term expression and integration into the genome, which can deoxygenate it again into your genome. transmission to the germline, passage into the sperm, and so many other genotoxicity and other things. All these studies were bypassed because we wanted a vaccine, and it was not considered as a, like a DNA altered medicine. It was, so they should follow the guideline, what is known as Gene Therapy Products Guidelines. So GTP guidelines was not followed for these vaccines. They are very quickly passed. It was required for the timing, but now it's time for the scientific community, those who are expert in this area, to work on this, find out the long-term, side effects of this molecule, especially for the next generation of people, generations of people. And what we have taken is adenoviral vector DNA vaccine. That also has problem because it has been shown that this is clumping up with the platelet factor PF4, which is strongly cationic, and this DNase, that is the vector DNA that we are using, highly negative. It is forming the cluster and leading to the formation of immunothrombosis. So blood clotting takes place and many other complications are there. So hindsight, if you think, maybe all of us should have taken the, what do you call, not Covishield, but the Covaxin. Because that's the time-tested vaccine, which is like a full attenuated viral vaccine. That should be safe. That will be also applicable for the wide range of immunity that you can get. Our Prime Minister was more intelligent because he took the Covaxin. So many of us have taken, I think, Covishield. So in future, you have to take the booster dose. Maybe some movie was made. I have not seen the movie. Some people say there's a movie called Vaccine. And that was made on the Covaxin development and all those things, which was made by this Hyderabad company. So what is needed to be done is for all these RNA therapeutics is like a long-term safety compliances should be followed to investigate their expression, integration into the genome, whether it gets genome integrated or not. We have to be careful. Transmission to the germline, passage to the sperm, embryo, and fetal and parental toxicity, and all this toxicity, genotoxicity studies, and all other studies should be done very carefully, at least now, to understand. Because now, RNA-based vaccine is good, because it's technologically good. It can be made very easily for any of the new sequence comes. Once you know the sequence, you prepare the corresponding RNA and follow the same science that has been done to develop this RNA vaccine. For cancer, it is being followed by, a large number of companies are putting billions of dollars to find out. Instead of, nowadays they get a lot of immunotherapies for the cancer patients, all the MAB, monoclonal antibodies and all those things. But slowly people are thinking that they can give RNA-based therapy for the cancer patients, but they have to be careful. And similar safety studies also required for what we have taken, like a DNA vectored vaccine, and also the small molecule vaccine, drugs that we are using, like a remdesivir or molnupiravir, which are working, developed as antivirals. So these kind of molecules we have very dangerous to play around. So what is known as like you reap what you sow. So we have to be responsible for what we make, and it can alleviate the disease for about the long term. We should not hide the facts and be very careful for the safety studies of this molecule. And I thank all my students. I have only 4 students shown here who are yet to be finished their PhD, all the funding agencies and people whom I have collaborated with. And this is my small group now, and this IISc, and thank you for your patience.
No question? Okay, okay, please.
But sir, is there any problem with the folding of that chain?
We don't know what is the 3-dimensional structures of these molecules. What will be, because as I said, like my students left, I don't have the molecule to do any binding with the RNA or DNA, and we hope that it can bind possibly, but if it folds too much, like a protein and all those things, it will have difficulty. But we don't know. I mean, sometimes it is like a binding driven, like a change of the conformation also can take place, because sometimes some of these conformations are flexible. If it has a better binding by not folding too much with a DNA counterpart and all those things, then maybe it will bind well. If there is also one issue about the charge compatibility, so that we can take care of by introducing some polar groups at the end to compensate for the charge. We can make, for example, solid phase synthesis of the peptide become easier for us because we can introduce some amino acid which has an amine group. We can make it cationic. Similarly, we can make it also anionic. So these studies we have not done, what kind of conformation it will take, whether it will bind to DNA or RNA, but yet to be explored.
Another question is, what was the cocktail solution for the global protection by using the cyberamide resin?
Which protection?
The cyberamide
FMOC chemistry we have done, and at the end we had to be clipped from the resin. So it's an ester linkages. And with just 1% TFA, it could, I don't remember exactly the functional group, but it's an ester linked. And then it could be clipped with just 1% TFA. Yeah, thanks.
Any more question or doubt? No? Then this is a time for vote of thanks. Okay. The Honorable Professor T.K. Chakraborty, Professor of ATE, respected Secretary, all the dignitaries, my colleagues at ICT, and dear students. Good evening. Today we have assembled here for the Professor K.K.G. Menon Memorial Endowment Lecture. This is a great learning for me with mind-boggling informations, and it is an eye-opener too. So thank you very much, Professor TKC, for this lecture, and I hope everyone enjoyed it thoroughly. Brief about the lecture that he spoke about, that mRNA-based therapeutics, the small molecule modulation of mRNA, and various oligonucleotides-based mRNA potential modulation for mRNA, cyclic dinucleotides analogs, and also he spoke about that COVID-19 vaccine based on mRNA, and then possible repercussion, that how it is getting accumulated in our body, and then long-term effect. So, and then in 2023, Nobel Prize in this medicine was given to Professor Kathleen Carrico and Drew Weissman, and jointly sharing the award for their groundbreaking this discovery of nucleotide-based modifications that enabled the development of effective mRNA vaccines against COVID-19. And also possible that repercussion that are mentioned here, that mRNA, the spike proteins are lethal in nature, and it is getting accumulated in the body because it cannot be broken down, and injecting in the body will lead to unbridled production, potentially lethal protein accumulating in the body for uncontrolled duration of time. Various research studies also said that it will be circulating in the body and accumulating, and potential result is the thrombosis. And one physician, Dr. Peter McCullough, in European Parliament expressed his concern, stating the fact that the recent development in heart attack or arrest causing because of these vaccines. And there are various results indicated that these vaccines causes myocarditis. So that means it clot blood in our body and causes low blood pressure and followed by heart attack. And various other heart inflammation or organ inflammations, and also cause artery fibrillations. So and the huge number of, montage of this cardiac arrest in young individuals have been recently reported. So in India also, we have witnessed some of the young, healthy people, youngsters, celebrities, they are collapsing while performing either dancing or playing cricket or practicing in gym. So is it regarding this effect of the these vaccines? So the question arises, who is next? So is it time that you, me, or any of our loved one, who is going to be the next person? So very intriguing and concerning fact. With this small note, I'd like to I express my vote of thanks. So it is my honor, privilege, how to propose a vote of thanks and acknowledge the contribution of all those who helped to make this event successful. First and foremost, I would like to thank the family members and friends of the late Professor K.K.G. Menon for their generosity, kindness, and contribution to the endowment. I thank the Honorable Vice Chancellor, Pandit Sir, for his encouragement and support to organize this event. Most importantly, words fall short to express our gratitude to Professor TKC for accepting our invitation despite his busy schedule and finding time for the lecture. Thank you, sir, for enlightening speech and in sharing your experience, your thought regarding this mRNA-based therapeutics. I would also like to thank former head of our department, Professor P.M. Bhatte, and present HOD, Professor Shekhar, for taking initiative and organizing this event. I owe special gratitude to Professor Sankarling, Dr. Som, Dr. Gary, Dr. Saha for their contribution as well. I would also like to express my heartfelt gratitude to one and all present here, all the students, delegates, for making this event a great success. Once again, thank you all for your kind attention. Thank you very much. Oh, so please join us outside for the tea. Tea is served outside. Thank you very much.